Yeah, I'm recording now and then share screen. Right, so let me start over. So today's talk is, to really learn how to write a good manuscript, right? And the only way to write a good manuscript is to really understand what is a good manuscript, or how do we know what are the components of a manuscript. And often you have a good faculty member as your mentor. You do learn how to write um, with them. Some mentors will rather write it themselves because they just don't have the time or the bandwidth to teach you how to write. And when I came to this country, like most of you, English was my third language. And obviously, I've come long ways. You should see my first article that I wrote, which my professor bled the whole paper, right? Handwritten red marks. So over time, obviously, we learn how to write better papers. For most part, I have a very high success rate in getting published. I can simply count with my hands less than 5 that got rejected or re-reviewed, but actually eventually accepted. And there is a reason for it. Right? So that with that credential, this talk we actually, I put work on this particular lecture in collaborations with my co-editor-in-chief, Norman Fry. And the reason we did this is because we were tired, really tired of getting articles from authors that are so poorly written. The science is good, but the delivery is bad, and we don't have time to go fix somebody's paper with little to nothing to gain. Why should I work on your paper when I'm not even a co-author, right? As a friend, I can do once or twice, but I'm not going to do it all the time. So this presentation has evolved since we did this as a part of the society in 2017. And so today I'm going to give you a much more evolved presentation. So this is the outline of my presentation. And it includes some introduction. We'll talk about why publish. We talk about what we mean by authorship guidelines. And then we dissect the anatomy of the manuscript. We'll go through all the components of a manuscript. And then we will talk about submission process. And I'll tell you something about the back end process. What happens once you submit your article to the journal? What happens at that end? So I hope this will be not only educational, helpful, but also educational. Give me one. All right. So why publish? Should somebody publish, Tej?
Not me. Sorry, I'm not getting your questions.
So why should we publish? What's the point of publishing?
To get promotion.
To graduate for me.
To graduate for some of you, for promotion for the others, right? Here's what nice Charles Darwin's quote: "A naturalist's life would be a happy one if it had only to observe and never to write." But what happens when we don't write? We perish. Okay. If you don't publish, you don't get your PhD. If you don't publish, you don't get your tenure, you don't get promoted, you don't get a job. You'll be Mr. Nobody or Miss Nobody. So you want to become known in your field, and if you or remain unknown. And another reason to publish is to authenticate new scientific knowledge. You find some new finding, and there's only way to authenticate it is publish it, right? It's Even now with the bioarchive, it's as good as not published until it get published in the real journal. And of course, we want to make sure the research is reproducible. And even if we do the same research or similar research, reproducible. And so as a scientist, like most of us are here, we must not only do science, but is also write science. And writing science doesn't come easy for others, but it comes easy for some. But I think that writing for me is a recipe that followed. If you follow the recipe properly, you can bake a cake or you can collapse the cake, right? So putting the right ingredients together in the right amount and creating and baking it, and it bakes into a nice fluffy tasty cake. It doesn't happen if you don't follow instructions. You can't simply add 5 eggs and 3 cups of flour and 2 cups of water at your whims and fancies. So writing is almost like baking a cake. You got to get things right. When you do bad writing, even if you have good science, it's not only prevent, it will delay the publication and you will get rejected. Very often people don't tell you why they're rejecting you. They don't tell you, it's a great science, you're a poor writer. They say it's beyond the scope of this journal. That's a cop-out answer. So I don't want to explain to you why I don't like your article. And we just say it's outside the scope of our journal. And sometimes it is really true outside the scope of my journal. But most often we just pre-screen reject, right? We do what we call pre-screen reject. The writing skills, the education or the current way of teaching students and educating individuals, we're committed to teaching them how to do technical stuff. We don't teach them how to write. We often neglect how to communicate, and this group already got in a lecture as to how to do a good presentation, the oral presentation. But writing is also communication. It's communication of your work in a written form. So communication is an art form, and believe it or not, many good scientists are very poor writers. They don't have to be English speaking or non-English speaking. Language background has nothing to do with the ability to write good writing. And why? Because many of them have not had the formal course in scientific writing. So that's another reason. And then, of course, if you get all of these things right, you have this huge gap with vocabulary and grammar. And especially 99% of the people in this room, in fact, I would say 100% of the people in this room today are non-native speakers, which means you even speaking wise, you make mistakes. You don't even know, right? I want to get more better. It's one of the things I keep hearing from this group. How do you get more better? It's so grammatically wrong. Yet nobody tells you, hey, Abdullah, you're making a mistake. That's not how you should say it. But if you're in my lab, I would keep correcting you until you get it right, because at the end of the day, you represent me and you got trained by me. So even when you speak, you're making mistakes, and you got to learn how to figure out how to improve your English. Read English storybooks. Listen to local news, right? National Public Radio. Listens to how they construct their sentences, how they word, and and for me, my English improved because I read incessantly, right? I love to read at least 3 books at any one time. And reading is the best way to improve your language besides listening. But reading popular material like People magazine, this and that doesn't help. Yeah.
You're muted.
So when you decide to write an article, you can simply start writing, or you can say this article is going to go to scientific reports. The first thing you do is go and look at the authorship guidelines. Every journal will provide you a list of things they expect you to do in order for them to review your article. And I'm going to, and in these authorship guidelines, there is also two things that I need you guys to made aware of. There is a publication ethics committee. And this is, it's you know, plagiarism, cheating, and all that stuff get discussed here. It's called COPE. They send you, if you register with them, they'll send you cases to look at every week, you know, where plagiarism or some other disputes happens. There's also International Committee for Medical Journal Editors. Of course, I'm an editor-in-chief for a medical journal, and we can look about how to the code of conduct on reporting, editing, and publication of scholarly work in the articles. And so the first thing you do is you figure out, so once you identify with the journal you want to write, you want to look at authorship guidelines. And then some of the things that needs to happen. You want to figure out who are all the contributing authors. And some of you already had a wonderful experience of figuring this one out late in the game, right? So you always start putting the people you're working with, make a list early. And you also make a decision who is going to be the corresponding authors, because corresponding authors are also known as senior authors. That's more prestigious to be a senior author. In our field and most fields, the senior author is the last author in the paper. And sometimes articles can have co-corresponding authors. Like if I work with Dr. Mandal expertise in the computer science or machine learning, and my expertise on the biological science. So when the reviewers come back and have questions for two different sections, then I'm responsible for my component and he's responsible for his component. And co-corresponding author works fine. It doesn't matter who goes front or back, but that's perfectly fine. And then we will talk about how to decide who is going to be the first author. And that's not a straightforward answer to that. And then, usually the lead writer who's going to take the lead in writing, putting together the first draft is also the first author. And we will also talk about how do you decide whose name comes first, second, third, fourth. In a team of 10 people, we can decide the order of authorship. I will also go over that. Stop me if you have any questions. It's an interactive session. So the justification for authorship really comes from taking into account all these components. The components includes the concept, who came up with the idea of the paper, like who really came up with the molecular mimicry, right? Or the design of the experiments, who designed, then who supervised, who provided the resources, who paid for your salary, the student TA-ship, right? So the person who provided those resources has to be there. The material, who, if it's especially in the bench work, we need a lot of material, and we need to know who are those persons. Who collected the data, who processed the data, who analyzed the data, who interpreted the data. Most students can process the data, they can't interpret it. They need somebody else to interpret it. And then this person has to do all the literature search. What is all the background for writing this article? And then who is writing? But even after writing, who spends all the time to critically reviewing your writing? If I'm going to spend 6 hours on your article, should I not be a co-author, right? You can question that. But if you are using colleagues for critical review, and I have decided moving forward, I'm not going to critically review any articles where I don't have a skin in the game, because it is a lot of time for me. And as many of you know that I'm very carefully provide feedback. And then even after all that stuff, if you don't submit your paper, then it goes nowhere, right? So we also need to know who is doing the submission. But the thing that really makes a huge difference is who is going to be the first author. That will be decided by this group of criteria. Who collected the data, who processed the data, who analyzed, who interpreted, who did the literature search, and who wrote. And all the other authors will be after that, right? So that's how we have to think about who are authors of a paper. So here is an Excel sheet that I use when I write papers with my students. I have all those components, and it describes the components, and you can literally put there, Mandal, Tej, Tanvir, and you start putting asterisks, right? From who did this, who did that, and then you just count the number, right? And then the order of authors based on how many access you have in these boxes. And in fact, you can say, right? And for example, here, if I count all the access, so here is one person who's contributed the most, and then you can see this person is my first author, this person is my second author, and so forth, and so on and so forth, right? So you can graph it, or you can just, if it's not too many, you can just eyeball it, but certainly you could graph in terms of who should be in what order the authors are. So it's and if you do this, there's never going to be unhappiness between your collaborators. You always make all these discussions and things before you start the project, not after you write the paper. So the authors should agree to be, if they're going to be co-corresponding authors, or they're going to be two first authors for equal contribution. Let's say Praveen and Vitali contributed equally to the paper, then they're going to be first and second author, but we'll put a little asterisk and say equal contribution, right? And then we agree with the order of the authors, and then all the authors, doesn't matter who is the first, last, or senior, they're all accountable for all aspects of the work. They have to read the paper, make sure that there's nothing there that they disagree. And as many of you know who have published, at least some of you who have published know that if you put the name of this author, they won't move forward on the paper until you approve of the submission, confirm submission. And in most journals, and almost all journals, the corresponding author acts on the rest of the author's behalf, and throughout the editorial review and publication process. And let's say, for some reason you forgot one of the authors, or the paper comes back and you have to do a little bit extra experiment. Now, Abdullah is graduated, so I'm going to take Imam to do this little bit work. And what happens? Now you want to add Imam's name on the paper, you must get consent from all the authors saying that I approve adding this person as an author. Correcting authors it's not a simple process. That's why from the very beginning, you make a list of all the individuals who are working with you. All right, so now let's talk about informations or instructions to authors, right? So I see still everybody has their camera off. I constantly check, right? So I multitask. All right. So we now, oops, it went too fast the other direction. For example, if you're going to publish in my journal, Journal of Medical Microbiology, if you click on this link, and it will give you the information to authors. All journals, any respectable journal will give you all the expectations of what a manuscript should look like. Read it carefully. Sometimes those instructions are very hard to comprehend, and even for me. So what I do is I print out the latest copy of an article from that journal and just follow the style. You know, if this is where it always boggles my mind. My students cannot copy the style. And then like, they take look at this. And that's partly because you are not observant enough. And it's not just the current group, but always, it's always, there's very few students can follow instructions exactly. But hopefully it will change as you all mature and get better at writing. You have to look at the font they recommend. I prefer to submit my paper in the font of the final publication because that's what they look at. And when they print to compare your article, sometimes the journals tell you it's okay to use Times New Roman or Arial, and they'll tell you what size. They'll tell you whether it should be 1.5 or 2. They will tell you what is the spacing around the document, and then if we allow footnotes or not. And then they also will exactly tell you how you put your affiliation. If you have 10 authors and 5 institutions, the affiliations can be A, B, C, D, or letters or symbols. So you got to check what is the journal requirement. And here's what I meant by printing out an article and then look at.
Dr. Mathee,
Yeah.
There is a question from
In the chat.
Praveen, is the equal contributing author is also a first author if her name is in second place?
Yes. Either your first or second place, if your equal contributing authors is equivalent to be first author.
Yeah.
Good question. Thanks, Ananda. Please do monitor the questions. Would be nice. Yeah, I appreciate that. Okay.
Okay, my pleasure.
Yeah. So, so it's failure to follow instructions do irk the staff and the reviewers and the the editors. And if the editors are picky, they don't even get to the reviewers. And then the reviewers will say, you know, this guy couldn't care less to check what is the expectations, and he or she wants me to waste my time on this article. Reject. Boom, go. Right? So you want to be very careful. So here's what I was talking to you about. You know, you see the introduction is in all capital letters, right? And you see the spacing, whether there is a space between paragraph. And this article, it's a really bad one. I cut and paste, and you can see all the things are not accurate, but you want to see how it is organized, and you want to see how the methods are organized, you know, and the subtitles starts within the paragraph. It is not a separate subtitle. And then results and discussion has separate subtitles that are separated by a spacing between the paragraphs. So you must go and look and understand what is recommended. So even if you cannot follow the instructions to authors, at least print out an article and use that as your blueprint to write your article. So now let's go through the anatomy of a manuscript. And as you already know, we have, obviously we all know this, right? There is a title, abstract, summary, or abstract or summary, depending on the journal. Every article have introduction followed by, they either could be called the materials and method or just methods, results, tables and figures, discussion. Sometimes results and discussions are combined. And then you always give a conclusion or perspectives. And then the whole series of statements, including acknowledgements and then references. So we're going to go through one by one, and stop me if you have any questions. So the title, I would always say go there last, right? But because after you write the paper, make up some title, but you must revisit the title. And it's also always the title you go last if you're reviewing the paper. And the title should be comprehensive enough, descriptive enough, so that if I pick up the paper, I say, hey, I want to know about this stuff. So you want to convey to the reader what is the content going to look like, and especially focusing on the core content. And titles is not where you write a long, complete sentence. It's never a complete sentence. It's really a hanging statement, okay? And it's important that your title is descriptive enough because, as you know, the Google search, the search platform, not just the Google, any search platform uses the title, the words in the title to find your article. If you want more scientists find your article so that they reference you, you need to make sure your title is catchy, but not pedestrian. What is not pedestrian? Not something like somebody who on the street would come up with this title. So you have to balance, and no, almost I've never known a journal telling me how long the title should be, but I do know that, right? Because Nature reports requires us have only X number of words. So you have to also look at that. How many words do we need in the title, right? So that again, you have to look at the instructions from to the authors. So here's an experience I had. I wrote this article, and we wrote this, and I just noticed, I don't know why all my oops, sorry. What happened here? Sorry, guys. Technical difficulties. All right, so here is article we wrote. This is one of my first paper as a faculty member, and we wrote this article, and I called it umpire regulates virulence mechanism. And then the reviewer came back and say the only experimental evidence that umpire regulates virulence mechanisms is blah blah blah. I wonder whether the title accurately reflects the findings of the manuscript. So this was a reviewer comment. Fortunately, they didn't reject my paper. They just told me that I need to change my title. And in the end, that title turned into a really long title, right? The regulatory repertoire of Pseudomonas aeruginosa C. Beta-lactamase regulator umpire includes multiple virulence mechanism. So it became extremely long title. And Pseudomonas aeruginosa should be italicized. I don't know what happened to my formatting here. I was changing all my font last night, so it doesn't have to do that. Okay, so I'm going to walk you through a little mini project, and this mini project is basically how to build a title. And this is one of the favorite thing for people to hear from me. So let's say here is first question you ask before you come up with the title, and I think Amrita has gone through this exercise, is to tell me what is the paper about, right? So this paper studied whether X therapy improves the cognitive function of patients suffering from dementia, right? This is what the paper about. So the second question you're trying to answer is, what techniques, designs were used, and who or what is studied, right? So then we came up with, it was a randomized trial. It involved 40 cases from 6 cities in Japan, right? Very short answers. We're only going for short answers. So the next question you want to answer is, what were the results? Observed an improvement in cognitive function. In other words, this X therapy, they observed an improvement in the cognitive function. So these are the three critical questions you must answer when you're writing a paper. What is the paper about? What techniques and designs used, and who and what was studied? Like what data set you used, right? And what were the results? Now let's build our title, right? So now building a title, you first delete unnecessary words, right? No, first you're supposed to come up with, no, did I miss that component? Yeah, this picture blocks your faces block my things. So based on these three questions, here's the statement, right? This study is a randomized trial that investigates whether X therapy improves cognitive function in 40 dementia patients from 6 cities in Japan, and it reports improved cognitive function. All right, this is not the title. Now with all these three questions, now I have this, what should be all the information that should be in the title. So now we are going to walk through how to make this title for this particular paper, right? So first you remove unnecessary and repetitive words. So this is the same sentence, right? What are unnecessary and repetitive words? For example, the study is that investigates whether it reports improved cognitive functions, because X therapy improved cognitive function, it reports improved cognitive function, right? So we now remove that. So now we have randomized trial, X therapy, improved cognitive function in 40 dementia patients from 6 cities in Japan. And then this is why I rewrite that, right? Randomized trial of X therapy, improved cognitive function in 40 dementia patients from 6 cities in Japan. So the next thing you want to remove is non-essential words. So there are several ways to look at the non-essential words. One is, you do not publish which country you're publishing from. Do Americans put that this study is from America? There are many, many manuscripts from US. Do we have US on all the manuscripts? However, the manuscripts coming from Bangladesh or India or China or Europe, it will say from Japan or from, right? So we, in our journal, we tell the author, remove the cities, because the minute you put Japan there, what happens? Whoever is not Japanese is not going to bother to look at this paper. All right, so you sort of reduce the audience to this paper, even though this paper might be useful for any other community or it's in Japan. So we're going to do, and we also don't say how many patients we looked at, because that's not the place for here. So we remove 40, we just say dementia patients. Now rearrange what you just have. What you have, X therapy improves cognitive functions in dementia patients, or randomized trial, or you can say cognitive function improvement with X therapy in dementia patients. You see how we went from that 3-line to one single line, but capturing all the information necessary to make this title for this paper. So that's so much for building a title. And I recommend, and I do this with my students, every time they write a paper, they need to answer this question, and they need to come up with this, go through this exercise of deleting unnecessary repetitive words, non-essential words, and then rearrange to come up with a title that works. All right, any questions about title? No. Was that fun?
Yes, sure.
I always like doing that.
So you suggest not to put any numbers in the title?
Yes. And You don't even put the place of study because that comes under materials and method, right? The site and data set and all that. No, you don't put numbers. You just want to be crisp and you just want to say, you want to catch their attention. So somebody might think, 40 samples is too little. Why should I bother to read this study, right? And some people with the form, when there's two studies with 40 and 400, they're going to ignore and say, it's not going to be good enough. All right, so let's
Dr.
move.
Mathee,
Yes.
is there any preference over active
Voice.
or the passive voice in the title?
I don't think you really have it's preferably an active voice, and here you can see, right? Cognitive function improvement. The active voice would be cognitive function improves with X therapy in dementia patients, would work as well, right? And here we did remove the randomized trial because it's not necessary. If they interested in the finding, they will go look at it whether it's randomized or not. So usually you do a positive spin.
Okay.
I think that's probably the better answer. You go towards a very positive spin.
Positive spin. Okay.
Yeah.
Thank you.
Welcome, dear. Yes, Vivian.
I do have a question. Is it fine like we include race or gender in the title? Or
Say that again.
is it recommend to put gender or race in the title?
No, not at all. Why would you do that? You only want people of your race to look at it? No, you want everybody to read it, right? So you don't put gender, you don't put race, in fact, the only time you see a particular thing is like HIV patients, they will say, right? Or in patients with HIV. And obviously HIV is such a huge field. There are thousands of papers. You want the people who are searching for the paper should find the HIV, right? Nobody is going to look for this particular paper by typing a randomized trial. There will be just way too many, right? And nobody is going to look for, but definitely people are going to look for cognitive function papers. People look for X therapy. They will look for dementia. So this is what is so important. You focus on the title is crisp and also gets picked up when they do a Google Scholar or PubMed, whichever way they are looking for an article. All right, so let's move to abstract, or sometimes called summary. When most readers, after looking at the title, they're going to look at your summary or abstract. Should I even read this paper further, right? And the reason is, once the title is exciting, and then they want to get a little bit idea on how to, whether they should even read it further. So when you are writing a manuscript, that is abstract or summary is the last thing you write. You write your article, and then in the end, it's again answering these 4 questions. A small introduction, what is the background, what is the purpose, 3 to 4 sentences. What is the hypothesis or rationale? What was methods used? What results were found? What was conclusion, implication? One sentence, couple of sentence, right? Introduction, 3 to 4 sentence, hypothesis, 1 sentence, methods maybe 1 or 2 sentence, results maybe 3, 4 sentence, conclusion, 1 sentence, implication, 1 sentence. That's an abstract. And every abstract, whether they ask you to specifically demarcate, must have all these components in a good abstract. And here's an example of the abstract that we did. I'm not going to read through it, it will take time. And we have a couple of sentences giving the background, and then we aim a hypothesis, and we give couple of sentences on methods and what we found, what the conclusion was, and what the implications were. when you address all these components, you have an abstract in your hand, a good abstract in your hand. Okay. So the introduction is, of course, is something that introduces the speaker or the reader to your material. And you always start from broad and then get to your specific project, right? So it has to be enough information that I don't have to go read another paper to come and figure out what is this paper is talking about. And most often, the person who's reviewing your paper is not in your field. You got to understand that. You send a paper, he may be a physicist, but he is not doing a simulation, right? He might be doing some other biophysics or, a machine learning. They might be doing something else besides what what you're doing. So you must be comprehensible so that not only the reader understands, the reviewer understands. Of course, the editor doesn't read the introduction. They go by the abstract and the title, right? And then, so you got to find all the literature that is published for that paper. It not just state, and this is something that really, really gets my goat, is that so and so found this, so and so found that. Albert et al. Showed this. No, I don't want all that. I want to say, from what he showed, what did it mean to you? You got to critically evaluate, not giving me a list of things that this person did that, this person did this. No, but how does that integrate into your work? And sometimes I get tired, I don't correct this. And I get this a lot from many of colleagues here. So you got to not just keep saying it. And in fact, don't even say, Mondal et al. Said this and Stebliankin said this. No, make a statement of what he found and put Mondal et al. In the bracket. Right? So don't keep saying, and the students are notoriously will do that. They will take a paper and they say, okay, Sharma et al. Said this, Al-Mamun states this, say that, and they compile together. Here's a literature review. That's not literature review. You are writing one sentence simply based on the title. If you read it carefully, there'll be more to say about the article than beyond one line, right? And that's what we mean by critical review of the literature, the past literature. And not just read the abstract and move on, especially if you're writing a PhD dissertation. You got to get deep into that literature to understand the background and everything. Now, what I recommend is, and I did this as a PhD student, after reading every paper, I'll put the paper on the side and I write down about 200 to 300 words of what I think this paper is all about. And what about this paper that I liked and I didn't like, right? So now I have that section for that paper. So when I'm writing, I'm bringing all those written stuff together and integrating it. And you must, as a PhD student, do that, right? As we get to our age, Dr. Mandal and I, we read very quickly. We can scan and quickly look at the methods. We don't have to get deep into it. We could more or less tell you where this paper is going, and then I might go read the discussion and see if these guys are full of it or they really mean something. So the ability to scan an article in detail has to come, and it won't come for another 20 years for most of you. Until then, you got to get down deep into it and read it. Okay, even now, if I want to understand an article, I have to read A to Z. I may go read a background on it. So start reading papers and start writing up this one-page paragraph, half a paragraph, or of what do you think that paper is all about. In the end, I had a stack of papers. Of course, those days we printed the papers, and each article I had a little paragraph on top of it. Right? So then I don't have to go and wade through the rest of it. And then you use all this literature to frame your hypothesis. So usually the last paragraph of the introduction is, based on all these studies, these are the clear gap statement. You make a gap statement, and then we say based on this we hypothesize blah blah blah, right? And the important thing is you provide a balanced view. Certain area of research is highly controversial, right? Some people say Pseudomonas is good for plants. Some people say no, it's bad for plants. So there is clearly two views. And what you do, you read all these papers and make sure you understand why are they saying this. You read all these papers, understand why are they saying this. And you decide to come a balanced view, right? Your balanced view is that, they both are right in their own ways, but there is a medium of somewhere they could meet, and you could make that statement, right? And then the good starting point in terms of introduction is the title, right? That's why developing a title for your paper is so important, because the title can inform you what goes into your introduction, right? And the tense can be mixture of tense, past and some present tense, but it's not. It all depends on what you're trying to say. So here's an example of a title and how my student would go and figure out what are all the things that he needs to do. Again, all my italics is gone. I am so annoyed. You know how I get annoyed. Some of you know I get annoyed. So the question is, the title of this paper is the Regulatory Repertoire of Pseudomonas aeruginosa, AMC beta-lactamase regulator AMPAR, includes multiple virulence genes. So this is my title, right? Remember the title that guy told me to rewrite it? So what do I need? I need a paragraph about what is Pseudomonas aeruginosa. I need to tell them what is a beta-lactamase. I need to have a paragraph on AMC. I need a paragraph on AMPAR. And then I need a paragraph on regulation of AMC by AMPAR. And I need to talk about what is virulence genes, or what is virulence and what is virulence genes. So essentially, my introduction will have 7 paragraphs, right? Based on my title. So develop your title, comprehensive title. You can figure out what are all the things that goes into introduction. It is not different from what I told you for your presentation, right? I said in the presentation, based on the title, your slide has to follow your title and identify information for each slide. It's the same, right? So this is literally the last paragraph in the introduction, right? And which talks about, in this study, we explore how exposure to sub-inhibitory concentrations of clinically relevant antibiotics alters the resistance pattern in Pseudomonas aeruginosa, right? And then our studies provide, again, don't give away everything, but you do write like 2 summary lines as to what is this paper all about. The insight into how combination treatment with 2 classes antibiotics might influence the potency of individual antibiotics in the clinical setting. In addition, we show the clinical importance of transcriptional regulator AMPAR in the development of antibiotic co-resistance by analyzing AMPAR in multi-drug resistant clinical isolates of Pseudomonas aeruginosa. I can remember, I'm a little tired today. Somebody came to me yesterday, was it yesterday, day before yesterday, told me, Dr. Mathee, you give a treatment with one drug and the bacteria becomes resistant to different drug. Was that you, Christian, who told me?
I don't think so. I don't
Okay, somebody
remember saying
did.
that.
And I was going to say, obviously, you haven't read our paper yet. This is the work.
It's me. It's me.
It's you, Tej. Sorry, Tej. So we published a paper many years ago on this very point. And this is the paper. Work done by Dr. Kumari in my lab when she was there. So you treat people with one drug and then the bacteria becomes resistant to other drugs. All right, so now we go to materials and method. And like I told you, it is methods in some journals, material versus method. So this one should have the design. And I would say for many of the projects we do, your design can include your pipeline, right? You describe your design of your project in the matrix and method, and then you go through the data collection, you write down all the experiments, right? And how you collected it, and don't miss out anything. And you know, which software or what chemicals, what, you know, if you are a biology writer, if you use a particular machine, we even have to state where that machine was bought from in the bracket, so that how much details they want to see. And then the processing, we always used to have a section on statistical analysis. Increasingly in all my articles, we have another paragraph on bioinformatical analysis. And then we certainly, everything, all the methods have to have a primary reference. What does that mean? The method you use, the reference you use have to be the original paper, not because somebody else did the same experiment and I read that paper, so I'm going to put the author, the reference as that author. That doesn't work. Let's say you use a method, you know, we do that all the time, right? We take a particular algorithm and then we modify the algorithm. We write that what exactly we did and what modification we did that is different from the original. In same way, methods, we do that. And then how we designate the units and the concentration is very journal specific. And of course, method section is all past tense. Right? It's you finished. You did this, you did that, you did this. It's all past tense. I hope you all know what is a past tense. And then now we go to the results section. And again, this combined with discussion. This is again, it's a constant struggle. If that paper has the results separate and discussion separate, it's hard for people not to bring together. So you got to you got to be very regimental about the results sections. Only talk about results. You don't interpret, you don't discuss the results. If you do have to, sometimes one sentence, because it needs to flow with the next set of experiments, right? And so in a way, it's better to write R&D together. In other ways, it's not so easy to write R&D together, results and discussion together, right? Now, how do we write the results, right? You have to use a logical progression, not necessarily in the order of the experiments done. Sometimes your first experiment, your first results is the last experiment you did. You have to figure out what is the story and how are you going to tell the story? Where are you going to start the story? And its tendency is to write, oh, I did this first and did that. No, you figure out what is the logical progression. So if you have a pipeline, or if you have a chart to say that, okay, this results can lead to this results to this results, and you want to go logical progression. Tense, past with rationale, and present with current results. So everything before you start presenting your results, you start with the past and then go to present tense. And again, we said that we can combine it. And so within results, how do you write the sections of results? And sometimes students they create all these beautiful images and they're stuck. They don't know how to write. And I keep saying, write down, write down, write down. And then sometimes I just have to get my whip out, right? Or I tell them you can't do any experiments in the lab until you write the paper, right? So, and again, I told you it's a simple recipe, right? What is the recipe? First, introduce the purpose of the experiment. Second, you explain how was it done. And third, you explain what were the controls, right? It doesn't matter if it's a computational experiment or a laboratory experiment or simulation, right? You start with the purpose, how was it done, and what were you using as a controls, because you can say this, I simulated this, or I did this, this is the data I found, you know, but what are you comparing is what is your control? How do you know that everything you did should give what you're given? So you always have run a known system to make sure your code or your process is reproducible, right? And then the first thing after you give these three, you write down what was the outcome of the control. So for example, as predicted or as expected, the control experiment worked as predicted and it did X, Y, and Z, right? Once you do that, and then the reader will say, okay, they have the controls, they're getting the results as expected. So what is the outcome of the experiment? And this is so critical, especially in a bench work, right? If your controls didn't work and you do get some results in your bench experiments, who cares? Your controls didn't work. I don't believe your experiment anymore. So until you get the controls work, don't do the experiment. That's what I would say. Because you waste a lot of time and money to get the controls. If you don't get the controls work, then it's a failed experiment. And then results as you see them. The tendency, and even in a computer science writing, our outcome is shown in table 1. So you are too lazy to tell me what is the outcome in table 1? Or you don't know how to explain the outcome in table 1. Here is a string of antibiotic resistance marker, and here is a bunch of bacterial strains. Go interpret yourself. And I'm going, I don't have time for this. I don't care for your paper. So if you put things in a table or figure, you got to explain. You tell them to go look at the numbers in the table, but you need to explain that information in your results. And don't just ignore negative results or positive results because they didn't go according to what you expected. The more you face the truth, the easier the reviewer will be. Oh, they did notice this didn't work, right? And you can say, this particular experiment didn't work. And in many times, we had, my student had a tremendous problem, right? Because his control won't work. He went back and recreated the strain, which for us takes very long time. And then we learned that it is the case for this particular strain. And then we said, okay, we tried 10 different ways. We could never get this setup working. And then we can answer the rest of the thing, right? So interpret or analyze to the extent required for the transition to the next section. So just focus on what you see from the results. And here is again, a sample results. This is a short title, right? I'm sorry, I'm using my papers here. So Pseudomonas aeruginosa, Ampar promoter expression. This is results, not even a conclusion. It's a quick short title. So I have a couple of sentences to talk about the intro, rationale, and gap, right? And then blah, blah, blah. And then I say that there is intragenic region, And then I write a couple of sentences on what we did, right? This was an experiment, but not details. But all these are critical for understanding the results. And then look at what I say, as predicted, right? The controls had no activity. Then in the absence of inducer, blah, blah, blah. And then you put the table 2. You don't have to say table 2 has the results. No, you start explaining the results and put the table 2 or figure 2, etc., exactly where they are. Got it? Okay, so what is next? Any questions so far? So every paper has figures, tables, and like last week, we rejected the paper because it had poor figures. Of course, the authors were pissed, right? oh, it's a great science. So don't assume you can hide behind great science and provide bad figures. The journals really specify the quality of the figures, right? Pictures are always worth 1,000 words, like the sunrise outside my apartment. And how do you decide? Some journals,
Dr. Mathee,
yeah.
I think I have a question. My student also have that question probably. In our computer science paper, in terms of control, like what kind of things we say?
Because we usually, I know we the way our experiments are doing, the DAG versus, for example, right? Differential gene expression analysis. We are looking at certain group of genes. There should be some publication that shows that these genes are differentially expressed.
Okay.
So we need to go back and check our output is exactly what other people see, or we are doing something very different. And in terms of, and then the other area where the control is an issue is how we know that DAG-seq2 is working as expected. Is there a way to control for that?
No, I don't think so. Mashur, Tanvir? Any comment on that? What Dr. Mathee is asking? Because you two are using Dissect2.
I mean, thinking of a control is not an easy and straightforward for each experiment. And
yeah,
you know,
what I see for my case. Yeah.
Right. We need to think about it. But usually, like for example, the students are running bunch of programs, these workshop students, right? And one of them is genomic analysis. So what I have them do is they are doing genomic analysis of a known strain, and the output has to be what is expected. Then they can run their strains and say, okay, I'm getting what I'm expected of this strain. So whatever I'm going to get from my strain is I can trust. So that's your control strain.
I see.
Right? And in experimental is the same way. You know, we always have the positive control that we know it works. We have to have a negative control. We know it shouldn't work. And if these two controls work, only then we can believe rest of the experiment. So in a, you know, that's.
You're muted. You're muted.
Even in the analysis we do, we can spike it with genes that shouldn't be there, right? We can label certain group of genes and it shouldn't come up spuriously. That's something we can think about, Ananda, and we should think about that. Yeah. All right. And you know, it was very funny. Obviously, you know, we have been around, we have been publishing over for close to 40 years now, and the journals went away from figures because they wanted tables, because tables were a bit more concise. And now most journals are going online. They rather have figures than tables, because tables are simply ugly and hard to interpret. However, you can check, each journal might say, you know, you only can have 4 figures or 2 tables. So you have to take into account. And then the quality of the figure, you have to make it depending on, you know, obviously some of Dr. Chapakin's lab, you know, you make all those color figures. And again, when the paper print is big, it costs a lot of money to print in color. They charged us a lot of money, so we are often opted to do black and white. And even with figures, you know, if you decide to use somebody's figure, you have to ask permission, or if you use the figure as a template and you change things, right? And what we call them is adapt the figure. So you can say this figure was adapted from this publication. That you don't need permission. It's only when you use the figure exactly it is, you need permission. And here's an example of a good figure, right? Where the font is, everything is bold, the lines are thick, and so the idea is when this figure printed, it will be printed shrunk, right? It's not going to look this size. It's going to look 1/4 of this size. When you shrink it, will that figure still be legible? And that's the yardstick. And before we had all this fancy equipment, and the way my mentor used to do that is she'll make a big figure and she'll print it 25%. If you print it in a 25% size and I can still read the figure, then I have a good figure. And you can only do that if you use large enough font and you use bold to do that. And they also, when you do the figures as figures, you don't write a lot of things. You don't write what plasmid. You want to keep to what is the content of each lane. So don't worry about this. This is a gel image of one of my postdocs. It's all published, right? So, and then this, for like I told you, every journal has their instructions, and the ours is, what is the maximum printed size, the line thickness, allow for reduction, just what I just told you. And they even tell you what are the preferred symbols for graphs. When you do several lines on the graphs, you go from circles, squares, triangles, and diamonds, right? And these are very specific instructions. If you're providing sequence data, it will tell you 80 to 100 nucleotides per line so that they can print it and it would look nice. And what kind of format they want, TIFF, JPEG, whatever not. And you just have to check the journal and see what is the instructions. Right? And then in terms of labeling, like, figure legends, right? Do you put the complete name? Do you put short figure period or no period? Or if you put small a or big A, or multi-part figures, whether these are in this format or capital format, check the figures in the publication if you don't understand the instructions to authors. So you must check that. And the other thing is that, especially if you're writing your dissertation, your thesis, or even your manuscripts, the legend comes below the figure, right? It's always starts with a short title followed by instruction details. The figure legend should be sufficient that the readers don't have to go to the manuscript to understand what the figure is all about. That's how much detail you have to give in your figure legend, okay? You very briefly explain the experiment, what they're seeing, and then the figure is there. And it's at the bottom of your figure because it's very important, right? So here is an example of a figure legend, and it's a short title, right? Followed by what the experiment was done. And then this was a two-part figure, right? And you must in each figure tell how many times this experiment was done, right? These 3 different biological replicate trials are shown. They always, the reviewers will ask you to say, how many times did you do the experiment? Triplicates or quadruplicates? I forgot to tell you in your method section, you got to say that how many times the experiment was run. Right? And we do that in computer science too, right? We do 1,000 times, and get the answer. And I always say that at each iteration, if you can get a measure, right? And then if you plot the graph, at the end of the day, the graph will taper off. And when the graph tapers off, you have run enough reiterations that it's not going to make any difference if you run it 200 times versus 100 times. So we have to figure out how to measure the what you call computer science experiments which you run the machine. right? Unlike us sitting in the bench. So here is a table, an example of table. And the table again has to be very clean, strains name, genotype, whatever it is, you try to figure out how to make it concise and clean. And you can certainly create all these tables in these days. I am pretty good at it. I'm trying to teach my students. You can create a very nice table in Excel, and then you can put it on the document, right? And that's and I highly recommend take a tutorial, Excel tutorial or PowerPoint tutorial, even Microsoft Word tutorial, even if you know how to do them, because there are a lot of little tricks you don't know. And what about table legend? Table legend is again, it has to be concise. It has to be comprehensive without having to go to the text, right? But the important thing is that legend for the table is always on top of the table, not at the bottom, right? It's always on the top of the table, right? No details on methods, just describe the table, define the units clearly. And if there is some things you want to explain about the figure, everything goes as the footnote at the bottom of the table, not at the top of the table. The top of the table, the title is a short one sentence title, no explanation, okay? Here's an example. Here's a table 1, right? This is the thing. And then we mark this. You see this little cross sign, a double cross sign, a star sign, and then I explain everything at the bottom, right? So this is what a good table should look like. And it's no different whether you're in a computer science or biophysics, or doesn't matter. All of you should learn how to make good tables. I know all of you make lousy tables. I've already seen and corrected. And if you want sometimes to show you how to do a good Excel table, I'll do that.
So question.
Yes,
so table does not have a legend?
Table only has a title, no legend. These are basically explanation. This is called the table legend, actually, the first line.
Oh, just a one line.
One line. It's a very short line. We don't explain anything, but anything we need to explain about the table goes at the bottom.
I see.
Yeah. And sometimes I don't want to fight with people, I tell them they don't follow, I say, forget it.
The reason I'm asking, I think I saw some table with legend also. That's why.
Yeah, they shouldn't. They're not supposed to.
I see.
That's all poor training, and then some journals just chalega, they don't care. But
Okay.
most good journals, if science, they won't let you do it.
I see. Okay.
Science and nature, the reason the high impact factor is so high, they're so nitpicky. Every detail is done, right?
Yeah.
It's mind-boggling, and yet it is the authors want to just flaunt the rules, and then wonder why the articles get rejected sometimes. All right, so conclusions, right? So after you have your results done and you written up and you summarize, make a list of all the conclusions you have gotten after those experiments. And the reason is, hang on a second, okay? And sometimes multiple experiments can contribute to one conclusion, right? You do experiment A, B, C, and you end up with one conclusion. Or sometimes you have one single experiment, you come to one conclusion. Because when you make these conclusions based on all the experiments, they basically will become your subheadings for your discussion. Because one of the things that everybody gets stuck is how to write a discussion. And again, this is, for me, it's a simple recipe. What are your conclusions based on your results? 3 conclusions, 4th conclusion, doesn't matter. Use that as your subheading and start writing why this conclusion came about. So the recipe is very simple, right? and you combine the results to make conclusion. And again, here is very important to active voice, right? Each conclusion becomes a subheading. You start with the introductory lines, restate briefly what we knew based on the previous studies, blah, blah, blah. In this study, I did this, and this is what we came up, and what is the overall interpretation based on the past and present. This is, sorry, I apologize, I jumped here. So every discussion section starts with the introductory paragraph, the first paragraph. That introduces the project or the paper and brief what we know and what was done. Okay, same thing. And like a little mini conclusion. That's your first paragraph. Then you put your conclusion as a subheading. Before I go there, I'm still in the first paragraph. So here is AMPAR is the master transcriptional regulator, blah, blah, blah. This is my introduction. In this study, we showed there is a complex regulatory network and we identified blah, 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 right? So this is my first paragraph. Then I put subheadings. Each subheadings, right, have how did you arrive at the conclusion. Again, don't rewrite your results. You got to summarize your results there, right? You summarize the results and then that's what I mean by recapitulate, not repeat, right? And then ask the question, is this results, how does it agree with the published literature? I love when it doesn't agree, then you have something to talk about, right? So if it doesn't agree, then say why you think that your results didn't agree with the previous results and speculate why, right? And we know what kind of speculation. This is where you really get creative. It's not the same because they used a different software, because they're looking at apples and oranges and we are looking at apples and apples. So this is where you say why your results are different from them, right? And then you say, we can address the differences if the experiment was done this way or differently, right? And also you got to say what is the limitations of your own experiment. The writers think that editors or reviewers will somehow overlook some limitations. No, if a good reviewer will say this guy forgot the address or this guy is overinterpreting or underinterpreting. So just be honest and say, we recognize, the limitations of this study. We didn't have enough samples. However, nevertheless, I think our finding is interesting, right? And then you finish each paragraph with an overall conclusion. And you see, for me, it's just a recipe. You follow a recipe, you learn how to do the recipe, right? And I don't want to go through this in a long ways. Again, it has a short title, right? The discussion, that my short title. And then I gave the past, what happened here. And then I talk about how we addressed it and how the whole thing worked. And then I finished it off the control. Even in the discussion section, you need to put the table and figure numbers so people can go back and look at it. All right. Okay. And, this is where we say, the finding. So sometimes, what you find is somebody already found it. So you can say this finding concurs with previously found, right? And then something says that, this is our final conclusion. So you end the paragraph with the statement. All right, let's just skip that. Okay, here is, right? See, here is one where the results didn't agree, right? And we explain the inconsistency seen with the experimental model where the authors used a heterologous host E. coli to determine, okay, basically our results were very different from this paper. And then we said the reason is our experimental system is different. So you address the inconsistency right up front. And then in conclusion, after you summarize, you do it with a short paragraph, right? And you can make it very fluffy and talk about, and become creative. You can add, all the new things to do, and you can this is really a little fluff and potential new experiments. All right, so that's the anatomy of the paper, but doesn't stop there. You need to have acknowledgements. And we talked about everybody who provided who are not in the author list has to be acknowledged. The funding information, you have to have increasingly a statement that there is no conflict of interest. There's also ethical statements, even there is no animal work or human work, and you have to say that there is no ethical concerns. You have to have the title there in this paper. And the funding information is very straightforward, right? Every funding agency will tell you how to reference their agency and your grant number, right? And you have to say that grant A for AM for Ananda Mandal, dissertation year fellowship, BS. So you need to put the person's names at the end of each agencies. And you should add the assistantship, scholarships, and fellowships you received in your paper when you write the paper. And here is, just an example, right? This where the study was supported by this grant to KM, FIU teaching assistantship to blah, 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 right? And also we have to add a statement. The funders had no role in study design, data collection and analysis, decision to publish or preparation of the manuscript. So there's no conflict of interest between the funding agency and the research. Because sometimes, remember, companies pay you to do the work, and you got to be very open about such things. And I already said that, anybody who are not named authors have to be recognized, right? Expertise, time to study, and all the stuff. And usually personal acknowledgement before the funding agencies. And increasingly, all the journals want the people who are acknowledged to also agree to be acknowledged. Right? I want to acknowledge Dr. Giri, but Dr. Giri has to agree that I'm allowed to acknowledge him in my papers. And you have to make sure that there is no personal and financial conflict, right? And if there is a pattern in application, you need to add that, and you need to declare there's no conflict of interest. All right. So then the ethical statement, if you're doing animal or human work, I would definitely recommend that FIU has the IACOC and IRB. Here's the two links. One is to NIH, and obviously I do give this talk in Europe quite a bit. So I always have my UK link as well. And then for animals, the same thing, UK Arrive guidelines for UK scientists, and that's NIH guideline for animal research. And you have to, and when you do animal reporting, you got to indicate how the animals were euthanized and etc., etc. It's a bit more work. References, right? Again, every journal has their own thing. And every statement you make has to back it up with either your own data or reference, right? Cite all ideas and concepts and texts data, not your own. Use primary references, not somebody else's. Somebody said this, somebody said that. Oh, I read this statement in this paper and I know this is not that person's work. So, you'll piss off the reviewer if one of the person you're ignoring is the reviewer, right? And the statements and references should match. Just because I want references, don't add references, right? And this happens all the time, and don't do that, and definitely don't offend the reviewer or editor. I can tell you, an alginate paper comes out in my area, and that talks about comparative genomics or something. I will see if my paper is referenced, right? Because I know I've published. And if you're going to send it to my journal and don't even reference my article, don't you think I should be pissed? I would be, right? So in terms of reference management tools, we talked about a few the other day. We talked about EndNote and we talked about Zotero. And guess what? There's just plenty of them out there, right? This is, I knew RefWorks and I'd forgotten. RefDB, blah, blah, blah. There's as many here. And we know that Zotero is free, but there could be others might be easier. I know some of us in our team are doing Zotero and some of us are doing EndNote. So you can choose whichever you want and make sure you learn how to use it. So this is the 3 that I used to always recommend, but you can't completely rely on the software. The software makes mistakes, doesn't change it changes the, if it is a Greek letter, it will change, italics will change. So you have to, every time you bring in a reference, make sure you correct it in this, whatever software you pick, right? And you use, be consistent when you're referencing. Every journal will ask you certain reference style, the Harvard, Vancouver, or APA. You can certainly find online, or before I go there, depending on the journal, whether you put a period or not, whether you put your A before or behind. And then in this case, yes, Nair A weeks O, and then it's Nair A O weeks, right? And in this case, the A and O are not, there's no period. And it all depends on the journal. And the page numbering can change, can be full numbering or could be half numbering. And then the title could be sentence style or whatever style, it depends, right, on the journal again. So you should check the journal. And there are Scriber has referencing guidelines for every style. If you don't know what they are, it gives you how the in-text citation should look like, right? And if you go to this link, you will see, if you're one author, how to put in-text citations within the text, and then how it should enter in the reference list, right? And oops, where are we? So, okay, so then now you have written your paper, and I always start with this paper, a cover page. And what is the cover page? The cover page is the first page of your article, which has the title of the paper, author list, affiliations of the authors, and it also has a short running title, corresponding author, the phone number, keywords, abbreviations, and sections or subject category of your article, right? And sometimes the keywords and abbreviations can come after the abstract. How do you decide running title? Usually it's a limited number of words. It's very journal specific that shows up in every page. Like Journal of Medical Microbiology, we want it within 50 and 60 characters. Again, being catchy is not a priority, but clarity and accuracy is. You can use abbreviations in these short titles. All right, where are we? Are we getting pretty late? 10:30. We started at 9, right? It's supposed to be 2 hours, and I am getting pretty tired. Okay, are we all tired? Are we good?
No, we are. At least I am fine.
We are good.
Okay, looking good. This is what I don't like Zoom. I don't get the energy from the audience. When you're in person, they energize you. And now I'm like, seems like talking to the wall. All right. And there are a few other things you have to follow, right? The journals will have specific nomenclature. They will say this is how the genetic words has to be, and certain chemical names and all that stuff the journals will have. And not most, all journals want line numbering. Either each page starts with the first line or continuous. I prefer continuous so there is no confusion which line in which page. So you start from beginning to end. You always recommend an editor from the board of editors. Otherwise, it will randomly go to somebody you don't want to go to. All papers ask you to recommend a couple of reviewers, 2 to 3 potential reviewers, and cover letter saying that this article is not submitted elsewhere and this we're only submitting it to you, and explain why this article is right for this journal, right? And make sure the how you write it, the style and everything, you make sure it works, right? All right, that's it. You're finished writing the paper. Now we're getting ready to submit it, right? Are we getting ready to submit, Lavinia?
Yay.
Yay, okay. I love this slide. Rushing sloppy work to market is not agile, it's just sloppy. All right. And don't rush any of your work half written, poorly written because you're in a hurry to publish. All right. So always pause and make sure your written work is good. Right. So I love this thing. All right. Now, often the journals you can submit as a Word document, it will create a PDF. But if you're worried that in the process of PDF creation, it's going to screw up your formatting, you create your own PDF and submit it. And then, you know, the figures and images have to be certain DPI. Sometimes they say it can be lower DPI for review, but later you have to submit better ones. All authors, you got to have email ID for all authors. This was a pain in the butt for me because when I had students go away, I don't have their email authors ID. So when the students leave, especially Dr. Mandal, make sure you get their Gmail address, okay? They don't read the FIU email and we need their attention. So I get all the Gmail addresses these days. All signed and agreed to author and the order. And, you know, if you're providing any supplementary material for your manuscript, follow the instructions, the numbering and the content. There are very specific instructions. Okay, you're finished writing. You're all done, you're submitted, right? So it comes to the office. What happens? Do you think you say, oh, I want this paper reviewed by Dr. Mathee, who is an editor for this section. It doesn't go to Dr. Mathee right away. It goes to an individual who's no science background. All they do is to see whether you have dotted your I's and crossed your T's, whether you followed the basic instruction. Okay. So when you go there, there is what we call the pre-quality control, right? This is all what happens once you submit. In the pre-quality control, you can be, based on your language, inability to follow instructions, you can either reject or you can assign an editor, right? You decide where you want to go, right? Once the editor assigned, only then the editor gets request to review the article and the editor can sit on it, or the editor says, you know what, nah, I don't see this English language is poorly put together. They haven't followed all the instructions. Because remember, the pre-quality control person is not very critical. You can even escape that person, but you cannot escape your editor assignment. And they can reject your paper. And this process can take, this pre-quality control or pre-screen reject might take 7 to 10 days. This might take you a month, depending on how busy the editor is, right? So the editor then sends it for review, right? Assigns it to a reviewer, and the reviewers can sit on it. And okay, okay. And so the reviewer, again, you know, reviewers have to deal with a lot of papers. They have their own life. They don't get paid to do this. This is professional service, right? So they assign, and the reviewer sends the comments. The reviewer might not tell you that they don't like the paper. The reviewer tells the editor without informing the authors that this article really sucks. This author doesn't have controls. He doesn't know how to write, but the reviewer comments might just give you some comments, right? But at this point, the editor says, I'm going to reject, right? Based on the reviewer comments, it goes back to the assigned editor, and the editor says, you know, I don't want to waste my time on this paper. They reject this paper. Or the editor says, no, I'm going to send it back, right? And accept. It doesn't stop here, right? What happens is the author editor would say, okay, revise, right? Sends it back to the author. You revise it. It goes back to the assigned reviewer. And then the reviewer comments come back. And again, it goes through this cycle of editor decision, right? There is no guarantee that at this point, it's going to go in this direction to accept. It might also go to reject. If you don't address the comments, if you come back with the same mess, right? And the editor going to say, no, I'm not going to waste my time. I'm going to reject this paper. So usually you have 1 or 2 reviewers, sometimes 3, but mostly 2 reviewers. When one reviewer really shoots you down, chances are the editor will reject. But the other reviewer just shoots you down without giving you comments. We will go to the 3rd reviewer because we know that the reviewer didn't pay attention. So once you, then only it's not a straightforward process. It is a lengthy process, right? And
Dr. Mathee, you are muted again.
Myself again. I went back. Sorry. Okay. Yes, so this pre-screen reject, as I already told you, right? Compliance and language, right? And then the reviewer really looks at the originality, significance, and acceptability, and may suggest improvement if necessary. If the reviewer likes you, gives you a lot of recommendations. If it doesn't look like you, will give you little recommendations. You know, and so once it gets accepted, it goes to the publishing department. Now another group takes over the article and they typeset the article. And that's the group that will call and have you have problem with the figures, this and that. They will work with the authors. And then when everything is sorted out, they put the paper together and then they send you the Gali proof. The Gali proof is the manuscript just before publication. And the Gali proof, they give you 1 week for the authors to make sure that they didn't screw up anything during the process of setting the paper up, put the figure in the wrong place or some silly mistakes. So that is the Gali proof, and everybody, the authors will get the Gali proof to correct. And then, so that's the whole process. But let's say if you get a review from your reviewers, editors, or reviewers rather, right? How do you respond to reviewers? You can't say, I did it. Oh no, the editors are terrible. They didn't get it right, and you get angry. But you may get it angry, but it doesn't matter. If you want the paper published, you still have to go back with your tail between your legs, right? So you get it right, you could see your paper published, get it wrong, and it could mean rejection. So that's what I mean. Even if you revise it, if you don't put your response properly, you can lose the paper. So, you know, reviewers who care will give you good comments, right? So be gracious even if the comments annoy you. So I always say, read it, get angry, don't do anything, come back a few days later, address that, right? Acknowledge your mistakes if you missed something, but if the reviewer says something nice, you say, oh, we thank the reviewer for being so positive, blah, blah, blah. And you make an attempt to address their questions. It's not just like ignore them, but at least they know that you're trying, but if you must disagree, explain why, right? But make sure after you address the reviewer comments, you also make changes to the manuscript, and you have to track. Most often the journals want you to track the changes, right? I would say it's also another art how you address reviewer comments. You copy and paste each comment and answer them, right? And then you indicate where the changes were made, the line and page numbers, color code the changes. Don't just say done or addressed, right? If you don't respond to all the reviewers' comments, it may result in rejection. All right. So these are a bunch of links for you guys to look at. And that's essentially end of my presentation.
Thank you, Dr. Mathee.
Thank you.
Wonderful presentation.
Thank you.
So
So
why didn't you give this presentation 3 years back?
I don't know. Nobody
I-
asked me to. I present this. I presented 2 years back in New Zealand. I presented it in London, 2019. I presented it in London and New Zealand.
You see, you presented to other continent, not to us.
You guys didn't invite me to present. What can I say? A lot of people know I do that. I tell them, look, I can do this, but FIU hasn't quite appreciated this work I do. So if they ask, I'll do. They don't ask. They always ask me to do how to do a slide presentation and oral presentations. Yeah. I even presented in the University of Miami. University of Miami Postdoctoral Association had me twice over there to present.
It's a shame on FIU.
Yep.
Yeah.
Dr. Mathee, there's a couple of questions in the chat.
Okay, you want to summarize for me?
Well, they're quite concise. So Vivian asks 3 questions. So the first one is, what is the maximum number of first authors?
There is no maximum or minimum. Minimum 1, I guess.
Okay, her second question was that she notices that in some manuscripts, the names of the authors,
the number.
are followed by a subscript.
Yeah. No, it is basically to show the number. So like if you have 5 universities, each number belongs to 1 university. There's no other purpose. No. The third question is depends on the journal. Some journals ask you to ABCD. Some journals ask you to do symbols. Some journals will ask you to numbers. That's very much depends on the journal. Okay. So now I want you guys to write there. Now that I've spoken almost more than an hour, almost 2. So what was the highlight of today's presentation? And what was challenging? Imam, better write down. I gave you a free lecture today.
Imam, you should be lucky. You got it on your first, the moment you arrived.
Yes. I just came here and got that lecture. I think it will be very much helpful for me.
Yes.
Thank you, Dr. Mathee.
You're welcome, Imam. Arasu, you have published. What's your take? Arasu disappeared. So Vitali, what did you learn new here today?
Yeah, I learned that writing a paper is actually the recipe, because like before I was like just trying to figure out and write what makes sense. Yeah, so now I'm going to download the recording and like just revisit all the steps again. So it's like, yeah, like once you talked about it, like that you first construct the title, and then you basically construct the abstract from the title, and then you also in the paper you explain all of those things one by one, and this is how the paper shaped. So I didn't know that.
Right, it's the training. At the end of the day, it's the pedagogical training, and yeah, I'm glad that this was a very useful presentation for you.
Vitali was like me. Vitali came in 2018. I came in 2018. So,
Yeah.
so for last 3 years, we didn't get this thing. So I feel neglected.
I didn't get this for last 5 years.
I didn't know you existed, Praveen.
Yeah.
No, it's a pity Arpit is not here. And Jinal and that group was also not here. They are lost. We informed everybody. Yeah. Yeah.
Yeah, I really appreciate it, Dr. Mathee. Yeah.
Uh-huh. Thank you, Ananda. I appreciate.
Now it will be easy. It will make my life easy, lot easier than yeah.
You know, it's you know, I was lucky that my PhD mentor was an amazing PhD mentor. I hope I can see her, travel and visit her. But after that, you know, when I started writing, and I always go back to my first paper that I wrote with her. And I always ask the question, how did we frame this, right? And then it just occurred to me, it is so simple, you know, 4-step process, each one of them. It's a 4-step process. And it's, you know, it's not like cooking Indian food. You can throw everything and throw some coconut milk, it tastes good, right? It's literally a good, nice piece of cake, which means you got to abide by the rules. And if you don't abide by the rules, then it will look like, you know, your cake will fail, flop, it will sink in the middle, and all kinds of things will happen. But yeah. Good. So I think this is really good for the PhD students as they're writing. And anyway, so for the students in the workshop, you know, you got to write manuscript style report for your final report. So start working on it. You know, you have an idea, you know what you need, your introduction, and hopefully your mentors are already providing you some material to write some introduction. And materials and method you write as you make, every time you do one experiment, so to speak, right? You run a Patrick, you write down exactly what you ran, what is the reference and how it is run, and then what is the output. Don't wait till last minute to write. So what did you learn new, Amrita, here today?
Dr. Mathee, I mean, as usual, it was very informative, and I've heard this lecture like directly from you, like because you have guided me for 2 papers back to back, so you have explained all these things to me like in person, like multiple times. So it was refreshing. It was like, again, before writing the 3rd manuscript, it was like revisiting all those instructions again. It was very helpful. Thank you.
Good. And this recording will be available, so you guys can listen to it again. In fact, this is the first time this lecture has been recorded. So,
Dr. Mathee,
yes,
my students need this recording ASAP right after this meeting.
we'll put it in the Google Doc, and
Okay.
yeah, and Masroor and Abdullah should access it. Yeah. Good. Anyway, guys, so thank you for being here. Make sure put your note there. I will capture it. And I will see you guys 9 a.m. Miami time and 9 p.m. Malaysia time. Vitali, I hope you can come. That's AMR talk tomorrow.
Okay, yeah, sounds good.
Yeah, you registered, right? The JMM series.
Yeah, I registered, but I don't see any emails about the link. So I'll look at it again.
Really? Did you get a link, Amrita?
They haven't sent any link, Dr. Mathee. But
Yeah,
I registered like twice already. I'm not getting any instructions from the society.
Can you go to the website and see?
is that the JMM seminar series?
Right.
Okay,
You guys
I
gonna
think
try to check
yeah,
the
that's the
social.
link available in the WhatsApp.
Yeah, but that link is to register. Does it show you? Give me a second, guys. I will.
No, the instruction said that you will receive a link just before the talk, but we haven't received anything.
Really? That's weird. I will. Hang on. Give me a second. I will check. Stop recording.